Our fifth and final physiology lab for the semester is a lab on hematology. And hematology is the study of blood in all of its different components. In lecture, we haven't yet spoken about blood and all of the different components of blood. We'll get to that pretty extensively in the spring semester early on. But for right now, let's have a quick refresher about the different components of blood that we'll be studying in physiology lab today. Remember that blood is a type of connective tissue. We say that it's a connective tissue because it has very extensive extracellular fluid. And as a result of that extensive extracellular fluid, the cells tend to be really far apart from each other. One of the reasons why we're so interested in the study of blood is because blood, for all intents and purposes, is a fairly easy type of tissue to sample. It's pretty accessible, and blood goes into contact with all of the different organ systems of our body. Because of that, blood is a really good indicator of the overall health status of all of our different organ systems. If there's a problem with one organ system, usually there is an indicator of that problem present in our blood. So we're interested in hematology or studying blood because blood is very accessible and it's very informative. If we break down the different components of our blood, we can very generally see that as blood is spun in a centrifuge, it separates out into its distinct components. This large portion on the top of the vial here, kind of yellowish in color, this is our plasma. Usually, plasma contains about 55% of our entire blood volume, meaning that more than half of our blood is composed of plasma. And remember, plasma is the extracellular fluid of blood. In between the top layer and this bottom layer down here, which I'll get to in a minute, there is this skinny little white layer. That's called our buffy coat. The buffy coat contains leukocytes, which are white blood cells, and platelets, which are important clotting factors. We'll come back to those. This buffy coat comprises less than 1% of our total blood volume. The remaining portion of our centrifuge tube here is this red portion here. These are our erythrocytes. Erythrocytes, or red blood cells, comprise about 45% of our entire blood volume. And that number can change depending upon elevation, depending upon whether or not you're fighting an illness, depending upon training. Various physiologic environmental changes can change our erythrocyte composition relative to the entire blood volume. Erythrocytes, or our red blood cells, always spin down to the bottom of a tube here. So anytime our blood is put into a centrifuge tube and spun around really fast, the erythrocytes always end up at the bottom because they are heavy. The reason why our erythrocytes are so heavy is because they contain iron. That's that really important structural component that allows hemoglobin inside of those red blood cells to properly bind and transport oxygen. White blood cells originate in our bone marrow. They originate in our bone marrow, and then eventually they mature and they make their way into the blood as functioning cells. We'll speak more about that specific process in the spring semester. So let's talk a little bit more in depth about erythrocytes. Erythrocytes are red blood cells, as I mentioned earlier, and importantly, they contain the protein hemoglobin. Remember, hemoglobin is a tetrameric protein that demonstrates quaternary structure. So basically, there are 4 separate polypeptide chains that come together and form one cohesive functional protein. Hemoglobin, like I mentioned earlier, has an atom of iron inside of it. That iron, in addition to making hemoglobin very heavy and causes red blood cells to precipitate in the bottom of a centrifuge tube, in addition to that, Iron also is responsible for giving hemoglobin, and therefore our red blood cells and overall our blood, that red color. So iron absorbs all wavelengths except for red, and so that's what it reflects. And so the more iron there is in a tissue, the redder it appears. As we mentioned earlier in physiology lab, the shape of our red blood cells is extremely conducive to its function. Remember, red blood cells are disc-like in shape. They're very flat, which means that relative to the volume inside of the cell, there is a very great surface area outside of the cell. Remember, oxygen and other blood gases move into and out of our red blood cells by simple diffusion. One of the ways to most easily increase the rate of simple diffusion is to increase the surface area of the membrane. That's why red blood cells have that distinct disc-like shape. Additionally, red blood cells lack a nucleus in all major organelles, which is why they can maintain that skinny shape and not lose their overall function. As a result of not having a nucleus in major organelles, red blood cells are incapable of dividing and producing more red blood cells. If we want to make new red blood cells, it has to come from precursor cells in the bone marrow. The other type of blood cells in our blood are called leukocytes. Leukocytes, or white blood cells, play a really important role in our immune system. And very generally speaking, these blood cells can be categorized in two basic categories: granulocytes, meaning they contain granules or vesicles, and agranulocytes, meaning they do not contain granules or vesicles. Within the granulocytes category, there are three different types of white blood cells, or leukocytes: basophils, eosinophils, and neutrophils. Basophils are important because they stimulate inflammatory processes. Histamine plays an important role in basophil cells. Eosinophils are important because they limit the size and therefore scope of inflammation. In other words, they regulate inflammatory processes. Finally, neutrophils are a type of phagocytic cell. They're by far the most abundant type of white blood cell, and in being phagocytic, that means that they engage in eating foreign objects, kind of like a Pac-Man. Moving over to our agranulocytes, we have lymphocytes and monocytes. Within lymphocytes, these are a type of cell that play a really important role in immunity. And there are two different types of cells that are lymphocytes. We have both B cells and T cells. B cells play a really important role in humoral immunity. Another way of thinking about that is adaptive immunity. B cells are important because they produce all of our antibodies. Antibodies recognize specific protein tags on foreign particles called antigens, and in doing so, they signal those foreign molecules for destruction. B cells, anytime they are introduced to a new foreign molecule, they will create an antibody in response to that, so that anytime your body sees that foreign molecule in the future, your immune system is primed and ready to handle it. B cells, by the way, are the very basis and understanding behind all of our vaccinations. This is why vaccinations work. T cells are the other type of lymphocytes. These play an important role in innate immunity, sort of the immunity that we have inherently going all the time. T cells function most importantly by secreting cytokines to help destroy foreign molecules. And then our final type of agranulocyte is a monocyte. Monocytes are types of macrophages. So we have these in all different kinds of places in our bodies, our lungs, for example, and we'll talk more about that in the spring semester. But these cells are important because they too engage in phagocytosis, or engulfing and eating, and therefore eliminating foreign molecules. Other important elements exist inside of our blood too, and they are easily identifiable by using processes like histology. Platelets, which are these little plankton-looking objects down here, platelets play a really important role in forming blood clots. As soon as we have a tear in vasculature and we start to bleed, signaling mechanisms within the walls of our endothelium in our blood vessels trigger platelets to become sticky. This is in a positive feedback mechanism. Platelets become stickier and stickier, and that process facilitates the formation of a blood clot. Again, we'll speak more about that in the spring semester. Plasma, which we've spoken about earlier, is the extracellular fluid in our blood. This is an example of plasma over here. It has this distinct yellowish tint to it. Within plasma, we have lots of various elements dissolved and suspended in water, water being the solvent, and then all of these things being our solutes. Various proteins, ions, nutrients like free fatty acids and glucose, and waste products as well are all found within our plasma. The most abundant protein in our plasma is this one right over here. This is something called albumin, and albumin is really important functionally because it binds and transports fat. Remember, fat is a non-polar macromolecule. It does not get along with water. But our body, and our blood included, is primarily made up of water. So if we eat a very high-fat meal, let's say a bunch of French fries from the state fair, and we have a bunch of fat brought into our system, we're still able to transport that fat around our body because albumin and other proteins like albumin can bind and transport something that otherwise would repel away from water. Our goals for lab today are to use Histology Time, a program that we used earlier in the semester, as a guide to look through lots of different histological slides of blood cells and all of the different blood elements. In using Histology Time, it's important that when we leave lab today, we are comfortable identifying blood cells and distinguishing between different types of blood cells and different types of elements. Another important metric from today's lab is to make sure that we're comfortable quantifying metrics of hematology. These can have really important clinical and research implications. For example, if I know approximately how many red blood cells are in a certain volume of blood, I should be able to calculate how much oxygen is in that certain volume of blood. And then I should be able to extrapolate that out to the entire body, how much oxygen is being carried in all of the blood volume in all of our body. Those are the types of metrics we want to be comfortable working with today. In other words, dimensional analysis, but in the context of blood physiology or hematology. Good luck.